Automation at early stage biotechs can be a real challenge, and there's some real technical and sort of process hurdles that you have to overcome. Principally, true early stage development in the space is really just that, development. Unlike other more mature industries, cell and gene therapy manufacturers are often working to identify optimal manufacturing systems and processes during early clinical work. In traditional biopharm, where your manufacturing processes are modified to optimize the output of a single small or large molecule, you can drop the API into an automated system in which you already know the reactor scale, the connectors, the purification scheme, etc., and scale up directly from the work your development team already completed. For biotechs, establishing a traditional fixed automation model means a very high investment in capital equipment and facilities that may not match the end manufacturing needs or even the clinical demand on the other side of product registration. Once set up, there may be no way to modify the process, and now you're stuck with a suboptimal product or process. The biggest strategic and technical advantages for automation really boils down to one thing: control. GMP manufacturing is really based on the idea that your process will result in the same thing every time. A quality product, by definition, is one that meets the established specifications every time. We know there's inherent variability in biology that we can't yet control, and we really only understand a limited set of variables that we should control. So when there's already that much variability and shift within the therapy itself, a reproducible process becomes the only way to meet the expectations of high quality GMP manufacturing. The quality benefit, the principal benefit of implementing early, is essentially an improved likelihood of clinical success based on fewer variations in the final product. Strategically, we know there are a lot of groups working on very similar indications, targets, and honestly, using very similar processes. In that context, bringing in well-controlled processes could be the differentiator to attract potential investigators looking to scale up and commercialize rapidly. A more robust process is just simply easier to transfer and carries less risk of failure. Yeah, the changes needed to implement new processes and automation can be a real barrier for people who have moved down the development pathway. So at a basic level, our vision is to develop processing and handling tools that generally fit into existing processes. Our new system, the Signata CT5, is really intended to be a connection between existing unit operations, allowing users to implement closed, repeatable versions of their process for moving fluids between operations. We've started with formulation and fill because that fits well with our existing product portfolio, but it really is going to be more than that. We believe that allowing users control to define the critical up or downstream unit operations instead of locking them into our consumables and processes allows them to integrate without a complete overhaul and revalidation of the entire manufacturing scheme. Adding in the ability to control parameters like volumes and flow rates and the flexibility to specify the desired outputs minimizes the changes from any manual processes. We want to work with the customers to help integrate our system into their needs instead of helping them change their processes to fit our system. So the next automation challenges for technology in this space, I don't think that you can answer that question for the industry as a whole without looking at the differences between the needs of allogeneic and autologous paradigms. Conceptually, once we get to the point of larger scale allogeneic manufacturing, the automation solutions for those products are very likely to be unique, purpose-designed facilities, not too different from the existing biopharm model, I believe. For allogeneic programs, I think the big challenge is really the same as it's been up to this point, and it's getting the biology right. For autologous therapy programs, on the other hand, I think that realizing the true promise of using cells or targeted gene therapy to address the myriad small population diseases will really be accomplished only through flexible and distributed manufacturing and licensing, or on-site authorized drug development processes at translational and high-quality academic medical centers. To that end, the next challenge in my mind is really the development of systems that allow easy, almost plug and play unit operations to be integrated and changed as needed for a specific product or therapy. Automated equipment should be developed that supports these multiple product specific operations and include the QA and QC systems to help ensure the reproducible manufacturing. This will allow flexible therapy designed for individual patients under the same controls and the same processes that are really expected for licensing of drug products.